Everyone, welcome to Module 9.2.1. And here we're going to have an introductory to some of the sensory pathways in the somatic nervous system. So keep in mind, we did the general overview in 9.1 and 9.2.1, 9.2.2, and 9.3 will be more specific about these things. So starting with some of the sensory information, there are some afferent divisions of the nervous system. So those include the receptors, the sensory neurons, the sensory pathways, and the efferent divisions of the nervous system, which include the nuclei, the motor tracts, and the motor neurons. So sensoreceptors are specialized cells or cell processes that monitor specific conditions in the body or the external environment. So when stimulated, a receptor passes information from the CNS in the form of action potential along the axon of a sensory neuron. Each axon is part of the sensory pathway. So the nerves, the nuclei, and the tracts that deliver somatic and visceral sensory information to their final destinations inside the CNS. So taken together, the receptors, the sensory neuron, and the sensory pathways make up the afferent division of the nervous system. Somatic and visceral sensory information often travels along the same pathway. Somatic sensory information is distributed to sensory processing centers in the brain, either the primary sensory cortex of the cerebral hemisphere or the appropriate areas of the cerebellar hemisphere. Visceral sensory information is distributed primarily to the reflex centers in the brainstem and the diencephalon. So in this module, we'll kind of consider some of the somatic portions of the efferent division. So the nuclei, the motor tracts, and the motor neurons that kind of control peripheral effectors, and whether somatic motor commands arise at the conscious or the subconscious levels. They travel from motor systems in the brain along somatic motor pathways, and which consist of motor nuclei, tracts, and nerves. So the motor neurons and pathways that control the skeletal muscles from the somatic nervous system. So as we previously noted, sensory receptors are specialized cells or cell processes that divide your central nervous system with information about conditions inside or outside the body. And we use the term general senses to describe our sensitivity to temperature, pain, touch, pressure, vibration, and body position. And you have general sensory receptors throughout your body, and they're fairly simple in structure. And some of the information that they send to the CNS reaches the primary sensory cortex and your awareness. And the CNS interprets sensory information on the basis of frequency of arriving action potentials. So, for example, when a pressure sensation is arriving, the harder the pressure, the higher the frequency of action potential. The arriving information is called a sensation, and the conscious awareness of the sensation is called that perception. So the special senses are olfaction, vision, sensations that are provided by receptors that are structurally more complex than those of general senses. So special sensory receptors are located in the sense organs themselves, such as the eye or the ear, and where the surrounding tissue protects the receptors. The information these receptors provide is distributed to specific areas of the cerebral cortex, so the auditory cortex, the visual cortex, and so forth, and centers throughout the brainstem. Sensory receptors present in the interface between your nervous system and your internal and external environments. Sensory receptor detects an arriving stimulus and translates it to an action potential that can be conducted to the CNS. So this translation or conversion process is called transduction. And if transduction does not take place, then as far as you're concerned, the stimulus doesn't really exist. So for example, bees can see ultraviolet light that you can't, and dogs can respond to sounds that you can't hear. And in each case, stimulus is technically there, but your receptors can't detect them. So speaking on the detection of that stimuli, how do we actually do that? So each receptor has a characteristic sensitivity. For example, a touch receptor is very sensitive to gustation, equilibrium, and hearing. And the to pressure as well, but are relatively insensitive to chemical stimuli. So a taste receptor is sensitive to dissolved chemicals, but insensitive to pressure. And this feature is called receptor specificity. Specificity may result from the structure of the receptor cell or from the presence of accessory cells or structures that shield the receptor cell from other stimuli. And the simplest receptors are the dendrites of the sensory neurons. The branching tips of these dendrites are called free nerve endings, and they're not protected by those accessory structures. Free nerve endings extend through the tissue in kind of the way that grass roots extend in the soil, and many different stimuli can stimulate them. For this reason, free nerve endings show little receptor specificity, And for example, chemical pressure, temperature changes, or trauma may stimulate free nerve endings that respond to tissue damage by providing pain sensations. Complex receptors, such as the eye's visual receptors, are protected by accessory cells and connective tissue layers, and these cells are seldom exposed to any stimulus other than light. So they provide very specific information. The area monitored by a single receptor is called its receptive field, and whenever sufficiently strong stimulus arrives at the receptive field, the CNS receives information. Um, and the larger the receptive field, the poorer your ability is to localize that stimulus. So, a touch receptor on the general body of the surface, for example, may have a receptive field of about 7 centimeters in diameter, and as a result, you can describe a light touch there that's only affecting the general area, but not an exact spot. On your tongue or fingertips, the receptive fields are much less than a millimeter in diameter. So in these areas, you can be very precise about the location of stimulus. So an arriving stimulus can take many forms. It may be a physical force, such as pressure, a dissolved chemical, a sound, or light. Regardless of the nature of the stimulus, however, the sensory information is sent to the CNS only in the form of action potentials, which are electrical events. As noted earlier, transduction is the translation of arriving stimulus into an action potential by a sensory receptor. And transduction begins when a stimulus changes the membrane potential of that receptor cell. And this change, called a receptor potential, is either graded depolarization or graded hyperpolarization. The stronger the stimulus, the larger the receptive potential. So the typical receptors for the general senses are the dendrites of the sensory neurons. And in these cases, the sensory neuron is the receptor cell. And your receptor potential that depolarizes the plasma membrane will bring the membrane closer to that threshold. A depolarizing receptor potential in a neuronal receptor is called a generator potential. And specialized receptor cells that communicate with sensory neurons across chemical synapses give us the sensations of taste, hearing, equilibrium, and vision. And in these cases, the receptor potential and the generator potential occur in different cells. The receptor potential develops in the receptor cell itself, and the generator potential appears later in the sensory neuron. The receptor cells develop a graded receptor potential in response to stimulation, and the change in membrane potential alters the rate of neurotransmitter release at that synapse. The result is a depolarization or hyperpolarization of the sensory neuron itself. So, if sufficient depolarization occurs, that action potential will appear in the sensory neuron. So, whenever sufficiently large enough generator potential appears, action potentials develop in the axon of a sensory neuron. And so, the greater degree of sustained depolarization at that axon hillock, the greater the frequency of the action potential in the afferent fiber. And the CNS then processes and interprets the arriving information at the conscious and the subconscious levels. So, speaking of some of the interpretation of sensory information that arrives at the CNS is routed according to the location and nature of the stimulus. We emphasized previously that the fact that the axons are in the CNS are organized in bundles with specific origins and destinations. So along sensory pathways, a series of neurons relays information from one point, so the receptor, to another, which is like a neuron at a specific site in the cerebral cortex. For example, sensations of touch, pressure, pain, and temperature arrive at a primary sensory cortex, and visual, auditory, gustatory, and olfactory sensations reach the receptive visual, auditory, gustatory, and olfactory regions of that cortex. So the link between peripheral receptor and cortical neuron is called a labeled line, and each labeled line consists of axons carrying information about one modality or type of stimulus. And the CNS interprets that modality on the basis of the labeled line over which it arrives. So for this reason, you cannot tell the difference between a true sensation and a false one generated somewhere along that line. For example, when you rub your eyes, you commonly see flashes of light. So the stimulus is mechanical rather than visual, but any activity along the optic nerve that it creates is projected to the visual cortex. And you experience that as a visual perception, and the identity of the active labeled line indicates the type of stimulus. Where it arrives within the sensory cortex determines its perceived location. For example, if the activity in a labeled line that carries a touch sensation stimulates the facial nerve of your primary sensory cortex, you'll perceive a touch on the face. All other characteristics of the stimulus, so its strength, its duration, and its variation, are conveyed by the frequency and pattern of the action potentials. So the translation of complex sensory information into meaningful patterns of action potentials is called sensory coding. Receptors can be categorized by the nature of their response to the stimulus. And some sensory neurons, called tonic receptors, are always active. Other receptors, called phasic receptors, are usually not active. Phasic receptors provide information about the intensity and rate of change of a stimulus, and receptors that combine phasic and tonic coding can convey extremely complicated sensory information. So, adaptation is a reduction in sensitivity in the presence of a constant stimulus. You seldom notice the rumble of tires when you ride in the car or the background noise from an air conditioner, considering you've seen and been around it so many times. The reason is that your nervous system quickly adapts to stimuli that are painless and constant. Peripheral adaptation occurs when the level of receptor activity changes. So the receptor responds strongly at first, but then it's actively and gradually declines, in part because the size of the generating potential gradually decreases. This response is characteristic of phasic receptors, which are also called fast adapting receptors. For example, temperature receptors are phasic. And you seldom notice room temperature unless it changes suddenly. So tonic receptors show little peripheral adaptation, and so they're called slow adapting receptors. So pain receptors are slow adapting, and this is one of the reasons why pain sensations remind you of an injury long after the initial damage has taken place. Adaptation also occurs along the sensory pathways inside the CNS. For example, a few seconds after you've been exposed to a new smell, awareness of the stimulus virtually disappears, although the sensory neurons are still quite active. This process is known as central adaptation. Central adaptation generally involves the inhibition of nuclei along a sensory pathway. So, peripheral adaptation reduces the amount of information that reaches the CNS, and central adaptation at the subconscious level further restricts the amount of detail that arrives at the cerebral cortex. Most of the incoming sensory information is processed in centers along the spinal cord or the brainstem at the subconscious level. This processing can produce reflective motor responses, but you're seldom consciously aware of either stimuli or the response. So the output from higher centers can increase receptor sensitivity or facilitate transmission along the sensory pathway. And the reticular activating system in the midbrain helps to focus our attention and heighten or reduce our awareness of arising sensations. This adjustment of sensitivity can take place under conscious or subconscious direction. So you listen carefully, your sensitivity and awareness of auditory stimuli increase. Output from higher centers can also inhibit transmission along the sensory pathway. Such inhibition occurs when you enter a noisy factory or you walk around a crowded city street. You automatically tune out the high levels of that background noise. So receptors for the general senses are scattered throughout the body, and they're pretty simple in nature. And we can divide them into three types. Exteroreceptors provide information about the external environment. Proprioceptors report the position and the movement of skeletal muscles and joints, and interoreceptors monitor visceral organs and functions. So if we were to use a more detailed classification system, we can divide the general sensory receptors into four types by the nature of the stimulus that excites them. So notrioreceptors, thermoreceptors, mechanical receptors, and chemoreceptors. And each class of receptors has distinct structural and functional characteristics. The difference between a somatic receptor and a visceral receptor is its location, not its structure. A pain receptor in the gut looks and acts just like a pain receptor in the skin, but the two sensations are delivered to separate locations in the CNS. Proprioception, however, is purely somatic sensation. There are no proprioceptors in the visceral organs of the thoracic and the abdominopelvic cavities. So your mental map of your body doesn't include these organs. You cannot tell, for example, where your spleen, your appendix, or your pancreas is at that particular moment. The visceral organs also have fewer pain, temperature, touch receptors than we find anywhere else in the body. The sensory information you receive is poorly localized because the receptive fields are very large and may be widely separated. General sensations are widely distributed in the CNS, but most of that processing takes place in the centers along the sensory pathways of the spinal cord of the brainstem. So only about 1% of the information provided by afferent fibers reaches the cerebral cortex in our awareness. For example, you usually don't feel the clothes that you're wearing or hear the hum of an engine when you're in a car. So pain receptors are especially common in the superficial portions of the skin and joint capsules, within the periostea of bones and around the walls of the blood vessels. Other deep tissues and most visceral organs have few pain receptors, and pain receptors are free nerve endings with large receptive fields. As a result, it's often difficult to determine the exact source of a painful sensation. Different nociceptors may be sensitive to extremes of temperature, mechanical damage, and dissolved chemicals, such as chemicals released by injured cells. Very strong stimuli, however, will excite all three receptors. So, for that reason, people describe very painful sensations, whether caused by acids, heat, or a deep cut. Use similar descriptive terms such as burn. Stimulation of the dendrites of a nociceptor causes depolarization, and when the initial segment of the axon reaches threshold, an action potential heads towards the CNS. So, two types of axons, the type A and the type C fibers, carry very painful sensations. Myelinated type A fibers carry sensations of fast pain or prickling pain. So, an injection or a deep cut produces this type of pain, and these sensations reach the CNS quickly, where they're often triggered by somatic reflexes. They're also relayed to the primary sensory cortex and so receive conscious attention. In most cases, that arriving information allows you to localize the stimulus to an area several inches in diameter. The slower type C fibers carry sensations of slow pain or burning and aching, and these sensations are caused by a generalized activation of the reticular formation and the thalamus. Therefore, you become aware of the pain, but only have an idea of the affected area. So, pain receptors are tonic receptors. Significant peripheral adaptation does not occur, and the receptors continue to respond as long as the painful stimulus remains. Painful sensations stop only after tissue damage is ended. However, central adaptation may reduce the perception of pain while pain receptors remain stimulated. And this effect involves the inhibition of centers of the thalamus, reticular formation, and lower brainstem and spinal cord. So, an understanding of the origin of pain sensations and the ability to control and reduce pain levels has always been among some of the most important aspects of medical treatment. After all, it's usually pain that causes us to seek that treatment in the first place. We usually tolerate or ignore conditions that aren't painful. And although we often use the term painful pathways, it's pretty clear that pain distribution and perception are extremely complex. So the sensory neurons that bring pain sensations to the CNS release glutamate and or substance P as neurotransmitters, and these neurotransmitters facilitate neurons along the pain pathways. And as a result, the level of pain experienced, usually chronic pain, can be out of proportion to the amount of painful stimuli or the apparent tissue damage. And this effect may be only one reason why people differ so widely in their perception of pain associated with things like childbirth, headaches, or back pain. And this facilitation is also presumed to play a role in phantom limb pain, which we'll get to in a little bit. So the sensory neurons may be inactive, but the hyperexcitable interneurons may continue to generate pain sensations. And the release of endorphins and enkephalins within the CNS can reduce the level of pain that a person feels. Endorphins and enkephalins are neuromodulators that inhibit the activity along the pathways in the brain. These compounds are structurally similar to morphine. They're found in the limbic system, the hypothalamus, and in reticular formation. And the pain center in these areas also use substance P as their neurotransmitter. Endorphins bind to the presynaptic membrane and prevent the release of substance P, reducing the conscious perception of pain, although the painful stimulus is still there. So temperature receptors or thermoreceptors are free nerve endings that are located in the dermis, the skeletal muscles, in the liver, and in the hypothalamus. Cold receptors are 3 or 4 times more numerous than warm receptors. So there are no structural differences between warm and cold thermoreceptors, and temperature sensations are conducted along the same pathway that carry pain sensations. They're sent to the reticular formation, the thalamus, and kind of to a lesser extent, the primary sensory cortex. Thermoreceptors are phasic receptors, and they are very active when the temperature is changing, but quickly adapt to stabilize your temperature. So when you enter an air-conditioned classroom on a hot summer day or a warm lecture hall on a brisk evening, the temperature change seems extreme at first, but you quickly become comfortable as that adaptation occurs. Mechanoreceptors are sensitive to stimuli that distort their plasma membranes, and these membranes contain mechanically gated ion channels. The gates open or close in response to stretching, compression, twisting, or other distortions of the membrane. And there are three classes of mechanical receptors. We have tactile receptors that provide the closely related sensations of touch, pressure, and vibration. And touch sensations will provide information about shape or texture, whereas pressure sensations indicate the degree of mechanical distortion. Vibration sensations indicate a pulsing pressure, and receptors involved may be specialized in some way. For example, rapidly adaptation in tactile receptors is best suited for detecting vibration, but your interpretation of a sensation as touch rather than pressure is typically a matter of the degree of stimulation and not in differences in the type of receptor that's stimulated. Baroreceptors detect pressure changes in the walls of blood vessels and in portions of the digestive, respiratory, and urinary tracts, and proprioceptors monitor position of joint and skeletal muscles. And they are most structurally and functionally complex of the general sensory receptors. So, tactile receptors. Fine touch and pressure receptors provide detailed information about a source of the stimulus, including its exact location, size, shape, texture, and movement. And these receptors are extremely sensitive and have narrow receptive fields. So, crude touch and pressure receptors provide low or poor localization. They give little additional information about the stimulus because they have large receptor fields. So, tactile receptors range in complexity from free nerve endings to specialized sensory complexes with accessory cells and supporting structures. So, free nerve endings sensitive to touch and pressure are located between epidermal cells, and there appear to be no structural differences between these receptors and the free nerve endings that provide temperature or pain sensation. These are the only sensory receptors on the corneal surface of the eye, but in other portions of the body, more specialized tactile receptors are probably more important. So, free nerve endings that provide touch sensations are tonic receptors with small receptive fields. Wherever hairs are located, the nerve ending of the root hair plexus monitor distortions and movement across the body surface. So when a hair is displaced, the movement of its follicle distorts the sensory dendrites and produces action potentials. These receptors adapt quickly, so they are best at detecting initial contact and subsequent movements. For example, you generally feel your clothing only when you move or when you consciously focus on a tactile sensation from the skin. Tactile discs or Merkel discs are fine touch and pressure receptors associated with sensitivity to shape and texture. And they're extremely sensitive to tonic receptors with very small receptive fields. So the flattened dendritic processes of a single myelinated afferent fiber make close contact with usually large epithelial cells in the stratum basale of the skin. Tactile corpuscles or Meissner's corpuscles give us sensations of fine touch and pressure and low frequency vibration. They adapt to stimulation within a second after contact or so. Tactile corpuscles are fairly large structures that measure about 100 micrometers in length and 50 micrometers in width. And these receptors are most abundant in the eyelids, the lips, the fingertips, the nipples, and the external genitalia. The dendrites are highly coiled and interwoven, and they are modified Schwann cells around them. A fibrous capsule surrounds the entire complex and it anchors it within the dermis. A laminated corpuscle or a Pansini corpuscle are sensitive to deep pressure because they're fast adapting receptors. They are most sensitive to pulsing or high frequency vibration and stimuli. A single dendrite lies within a series of concentric layers of collagen fibers and supporting cells, basically specialized fibroblasts. And the entire corpuscle may reach about 4 millimeters in length and 1 millimeter in diameter. The concentric layers are separated by interstitial fluid, and that shields the dendrite from virtually every source of stimulation other than direct pressure. Laminated corpuscles adapt quickly because distortion of the capsule soon relieves pressure on the sensory process. Somatic sensory information is provided by laminated corpuscles throughout the dermis, notably in the fingers, the mammary glands, and the external genitalia, and the superficial deep basque, and in joint capsules. Visceral sensory information is provided by laminated corpuscles in the mesenteries, in the pancreas, and the walls of the urethra and the urinary bladder. Ruffini corpuscles are also sensitive to pressure and distortion of the skin, but they're located in the reticular dermis. These receptors are tonic and show little, if any, adaptation. The capsule surrounds a core of collagen fibers that are continuous with those of the surrounding dermis. So in the capsule, a network of dendrites is intertwined with the collagen fibers, and any tension or distortion of the dermis tugs or twists the capsular fibers that are stretch and compress the attached dendrites. And alternating activity in the myelinated afferent fiber. So your sensitivity to tactile sensations may be altered by infection, disease, or damage to sensory neurons or pathways, and for this reason, mapping tactile responses can sometimes aid in clinical assessment. Sensory loss with clear regional boundaries indicates trauma to spinal nerves. For example, sensory loss within the boundaries of dermatome can help identify the affected spinal nerve. Tickle and itch sensations are closely related to sensations of touch and pain, and the receptors involved are free nerve endings. Unmyelinated type C fibers carry that information. So people usually, but not always, describe tickling sensations as pleasurable. So a light touch that moves across the skin typically produces a tickle sensation. Physiological factors influence that interpretation of tickle sensation, and tickle sensitivity differs pretty greatly from person to person. Itching is probably produced by the stimulation of the same receptor. However, we're unsure. Specific itch spots can be mapped in the skin and the inner surfaces of the eyelids and the mucous membranes of the nose. So, itch sensations are usually absent from other mucous membranes and from deep tissue and viscera. Itching is extremely unpleasant, as I'm sure you know, and even more unpleasant than pain. Um, Individuals with extreme itching will scratch even when pain is the result. So, itch receptors can be stimulated by the injection of histamine or proteolytic enzymes into the epidermis and the superficial dermis, and the precise receptor mechanism is pretty much unknown. So, baroreceptors monitor changes in pressure in an organ, and a baroreceptor consists of free nerve endings that branch within the elastic tissues of the wall of a desensible organ, such as blood vessels, or a portion of the respiratory, digestive, or urinary tract. And when the pressure changes, the elastic wall of that tract will recoil or expand. The movement distorts the dendritic branches and alters the rate of action potential generation. Baroreceptors respond immediately to a change in pressure, but they adapt rapidly, and the output along the afferent fiber gradually returns to normal. Baroreceptors monitor blood pressures in the walls of major vessels, and these vessels include the carotid arteries, the aorta, things like that. The information plays a major role in regulating cardiac function and adjusting blood flow to vital tissues. Baroreceptors in the lungs monitor the degree of lung expansion. And this information is relayed to the respiratory rhythmicity centers, which set the pace of respiration. Comparable stretch receptors at various sites in the digestive and the urinary tract trigger a variety of visceral reflexes, which include urination and defecation reflexes. And we describe these baroreflexes in the urinary and digestive systems in AMP2. Proprioceptors monitor the position of joints and the tension of tendons and ligaments and in the state of muscular contraction. There are three major groups of the proprioceptors. We have the muscle spindles. So muscle spindles monitor skeletal muscle length and trigger stress reflexes. You have the Golgi tendon organs. Golgi tendon organs are similar in function to the Ruffini corpuscles, but are located at the junction between the skeletal muscle and its tendon. In a Golgi tendon organ, dendrites branch repeatedly and wind around the densely packed collagen fibers of that tendon. These receptors are stimulated by tension of the tendon, and they monitor the external tension that's developed during muscle contraction. We also have receptors in the joint capsules. Joint capsules are richly innervated by free nerve endings that detect pressure, tension, and movement at the joint. And your sense of body position results from the integration of information in these receptors with information provided by muscle spindles, Golgi tendon organs, and the receptors of the internal ear. Proprioceptors do not adapt to constant stimulation, and each receptor continuously sends information to the CNS, and most proprioceptive information is processed at the subconscious level. So only a small portion of the arriving proprioceptive information reaches your awareness. Chemoreceptors are specialized nerve cells and can detect small changes in the contraction of specific chemicals or compounds. So, in general, chemoreceptors respond only to water soluble and lipid soluble substance that are dissolved in body fluids, so such as interstitial fluid, blood plasma, and the CSF. and these receptors inhibit peripheral adaptation over a period of seconds. Central adaptation might also take place, and the chemoreceptors are included in the general senses, do not send sensory information to the primary sensory cortex, so you're not consciously aware of that sensation they provide. The arriving sensory information is routed to brainstem centers that deal with the autonomic control of respiratory and cardiovascular functions. Neurons in the respiratory centers in the brain respond to the concentration of hydrogen ions, so the pH, and the level of carbon dioxide molecules in the cerebrospinal fluid. Chemoreceptors are also located in the carotid bodies, near the origin of the internal carotid arteries on either side of the neck, and in the aortic bodies between the major branches of the aortic arch. These receptors monitor pH and the carbon dioxide and oxygen levels in arterial blood. And the afferent fibers leaving the carotid or the aortic bodies reaches the respiratory centers by traveling within the cranial nerves 9 to the glossopharyngeal and 10 the vagus nerve.